Hello and welcome to Inside Exercise. I'm Emeritus Professor Glenn McConell. I just finished chatting with Associate Professor Abigail Mackey from the University of Copenhagen. She's an expert on muscle in regards to what makes up muscle, what happens with muscle injuries, the causes and the consequences of muscle injuries, the cells that are in muscle, muscle repair, muscle innervation, so how the motor nerves coming from the spine interact with muscle, effect of aging on muscle, the likelihood of injury with age, muscle innervation, so whether you can actually slow the loss of muscle fibers with exercise. We also talked about where muscle injuries tend to occur. So, depending on the muscle, is it happening mainly in the muscle fibers themselves, in the joint between the muscle and the tendon, or in the tendon? We also talked about different models that are used to study muscle injury and repair. So, often it's eccentric exercise, which is, the muscle contracting while it's lengthening. We also talked about some rodent models that are used. I think you'll find this really interesting, so stick around. Hi, Abigail, how are you? Thanks for coming on.
Yeah, hi, Glenn. Good morning here from Copenhagen.
Yes, sorry, I made it a little bit early for you. What time is it over there?
It's fine. It's
well,
8:45. It's not that early.
It's more that it's probably taking a while to get light. Hey, yeah, it's great to have you on. And as I mentioned before to you, before we came on, a lot of interest on Myo Twitter, flying around, having you on. So yeah, we're going to talk a lot about muscle injury, muscle regeneration, innervation, all that sort of stuff. Why don't you give us a bit of a background? How did you get into this area, or how did you even get into research to start with? And were you like an exercise type first and then got into research, or the other way around? Or
yeah, so my background is that I studied sports science, and I suppose like most of the people on that course, we were active exercisers in one form or another. But it's funny looking back on it now, because I ended up in the muscle regeneration field. And I distinctly remember actually a lecture in one of my undergrad courses on muscle repair. It was the only lecture we had, and it was only part of a lecture covering muscle repair. And it was a figure from Rick Lieber's, one of his books. A sort of hand-drawn schematic of what happens when a muscle fiber gets damaged, and the infiltration of immune cells and activation of muscle stem cells, and repair of the muscle. And I thought that was absolutely fascinating. And I was so disappointed to hear that the story was over after 30 minutes. And I think that was probably a moment that really defined my interest in research in muscle.
I think you got more than I did a similar, it was called Human Movement Science, so exercise science, 1986 to 1989, so showing my age. I'm pretty sure we didn't get half a lecture on that. But, so you found that interesting, and and here you are. But well, why don't you give it a bit more? So you did I think you did your PhD in Ireland.
Yeah. And then when we were looking for opponents for the PhD defense, I think we realized that I had most of the work I had cited, or a lot of it was coming from Copenhagen, from Michael Kerr. So we asked him if he would be an opponent and come to Ireland for my PhD defense, which he did. And then he offered me a postdoc after that. And then the rest is history.
So it sounds like he was
for a 2-year postdoc, and I stayed for life.
That sounds like he was impressed with you then.
It worked out well. I mean,
Michael Kerr, I know, is into the tendon stuff, and we'll be talking a bit about this sort of things, of course. But I actually knew him during my PhD for doing all this glucose, really amazing stuff. Looking at giving people epidurals and then electrically stimulating them to cycle and look at their glucose output from the liver and things like that. So he's been around for a while and doing great stuff. Yeah. All right. So why don't you give us some background first, so everyone's on the same page? So, why don't we think about what even makes up a muscle to start with? Because, we tend to do, or I tend to do in my studies, a lot of muscle biopsies, and then you grind it up and see what you get. But there's a lot of different cells in there and whatever. Why don't you just give us a bit of background?
Yeah, well, I suppose the important things for the context of my research that we're going to cover here is probably the connective tissue layers of the muscle, that it's important to remember that the muscle is attached to tendon, which is predominantly collagen. But there's also collagen throughout the muscle, surrounding the individual muscle fibers, arranging the fibers into, or organizing them into bundles. And then of course the outer fascia, the epimysium, connecting the whole muscle to the tendon. And then of course within this connective tissue, there are many different cell types. So this is where the blood vessels permeate the muscle. So there's a lot of endothelial cells, pericytes, fibroblasts, of course, to produce all this connective tissue. There are resident immune cells in the muscle, which are important for repair, and stem cells. And then depending on which population you're studying, there will be some adipocytes. Yeah, and I guess that's basically it, apart from the muscle fibers, the cells themselves.
Yes. So I was just thinking, I guess when, as I said, we do muscle biopsy studies, and it's probably okay if you're measuring glycogen and things like that, and maybe AMP-kinase and things that we've done. But when you start doing, say, messenger RNA, and you're amplifying up from this muscle, there's all these other bits and pieces in there that you tend to just sort of hope are not contaminating your samples. Yeah.
Yeah, and I suppose that's, I started off doing a lot of microscopy of muscle biopsies, and it was one of the things that really fascinated me, that you can have one cell type responding to a specific stimulus more than the others. I mean, they're all interacting with each other and influencing the activity. But it's true, it always hurt me a little bit to have to homogenize a whole muscle and measure a signal coming from that, and not really knowing where specifically those signals were coming from in terms of the cell types that were activated. But I think things are, it's quite an exciting time now for people who have the chance to work with muscle biopsies because of the technology that's available. More and more people now are starting to work with single muscle fibers to try to dissect differences in the responses between type 1 and 2 fibers, which I think is a huge advancement for the field. And also these single cell technologies where you can virtually analyze each cell, these different cell types that I talked about, the fibroblasts, immune cells, stem cells, whatever, analyze each cell individually and get a profile of what genes they're transcribing, as well as the myonuclei themselves. So it's a really exciting time, I think.
And I know you do, like, say if you take the biopsy, you do cross sections, and then you can, I know you do some microscopy where you look at, so you don't have to just tease. They say there's always a funny teasing the cells out. You don't have to tease them out. You can also do cross sections and throw your antibodies on there and whatever as well, right?
Yeah, and that is also cell specific. Then you can look at responses of one fiber type, or I do a lot of cell counting. So how many satellite cells are there, and does the number change with different interventions?
It's tedious work. So so if we start talking about, you've mentioned satellite cells a few times. So if we start thinking about when you injure a muscle, what happens there? So the satellite cells get activated, etc. But that's about as far as my knowledge goes. Why don't you tell us a bit about that, and stem cells or whatever? Yeah.
So I suppose satellite cells are kind of leftover stem cells from development, if you like. They are specialized in the sense that they prefer to make muscle. I mean, you can perhaps force them to make other things in cell culture, but basically in the human body, that's what they do. They will make muscle if it's injured. But their stem cell-like properties come from the fact that they can maintain their own cell numbers. So first of all, they're activated and they divide. And not all of those cells will fuse to become the new muscle fiber or fuse in with an existing muscle fiber. Some of them will go back to being satellite cells, so that you have satellite cells for your whole life. And we see that in muscle biopsies of very old individuals. People don't run out of satellite cells.
Okay. There was talk about that at one stage, right? I guess it was early in the understanding. It was like you'd sort of use them all up or something, but you literally regenerate. So what, each time you activate, what, they divide and you sort of use one and get another one, or how does that actually work?
Yeah, no, so you have one cell and it divides, and there are different theories, but one of them is that one of those new cells will go back to being a satellite cell, and the other one then will maybe fuse in with an existing muscle fiber to add a new myonucleus, or it will find another satellite cell to fuse with. And then when you have enough of those, you have a small myotube that can become a new muscle fiber.
Okay.
If a fiber has been damaged.
So we're talking about, just so people understand, again, because people that aren't in the field or haven't studied exercise science don't actually realize how many muscle cells there are. They might think there's 50 in your quads or something. Like, when you have a muscle biopsy, because I remember I had that tedious job as well of going through and counting them all and whatever. Just a normal muscle biopsy, which is like, I don't know, a rice grain or two, there's about 600 muscle fibers, right? And you're talking about regenerating, each stem cell is regenerating like one muscle fiber, yeah? Yeah,
but I suppose it depends what type of damage we're talking about, because there's some muscle fibers are obviously quite, that they're not only large in terms of their diameter, they're also very long. And that depends on the anatomy of the given muscle. So many muscles are pennate, but for example, some of the muscles of the hamstrings, you would have muscle fibers running the entire length of the muscle, so almost as long as your femur. So that's one giant cell, and there will be hundreds of thousands of nuclei in that one cell. So they get damaged and they can be repaired without being replaced, but there's some damage where the damage is so severe that that entire muscle fiber will need to be replaced.
Okay. So when you do muscle biopsies, we tend to say, and I have to say, sometimes I felt a little bit vague. The participant will say, well, what happens with the muscle? And you say, oh, it gets regenerated. So you're taking that out, and you just say it is your I know we don't biopsy the hamstring, but just say you've got this muscle running the length of your femur, and you're taking like 500 muscle fibers, little bit of it out from the middle. They will just all regenerate that spot using satellite cells.
Yes. So I think in the case of a biopsy, I don't know, but I guess that it's such a severe insult to the muscle fiber that the whole fiber will die, what's left of it in the body. This is one of the fantastic things about studying skeletal muscle, and the reason we can allow ourselves to take so many biopsies is because we know that the tissue does regenerate. So you have a process where, and we've studied this using electrical stimulation. So we used voluntary eccentric contractions. I think those have been around for decades as a model of damage, where participants do maybe 200 maximal eccentric contractions, the forced lengthening mode. And that in some cases induces severe damage. But if you use electrical stimulation to induce the contraction rather than voluntary, it seems we get a lot more necrosis of muscle fibers. So we've used this as a model of muscle damage and regeneration now for a while. And what happens there is that after a few days, you start to see basically a disintegration of the muscle fiber. So there's no more sarcomere alignment, everything's disorganized, and you see a lot of immune cells coming inside the muscle fiber. So this, one of the layers of connective tissue that I referred to at the beginning, the basement membrane surrounding each fiber, is preserved throughout this process. And that's quite important because this serves as a sort of scaffold, if you like, for the new muscle fiber to be formed. It also reserves that space in the muscle for this new muscle fiber to be formed. So these immune cells come in and they basically consume all of the dead muscle fiber material. And not only that, and that's one of the interesting things about this cell-cell communication, that some of these macrophages, especially the pro-inflammatory type, activate these muscle stem cells, the satellite cells, to proliferate, divide, and start to basically fuse at the same process as if any of the viewers have worked with cell culture, they'll know that you put some muscle cells in a petri dish, and they will fuse with each other to form these long myotubes. Yes. And this is what we see in vivo as well. And you have several of those then that will fuse with each other. And then eventually you have a very small muscle fiber that will grow back to its pre-injury size.
Yes, and then it's kind of protected, isn't it? So those classic studies where you do eccentric exercise to damage a muscle, and then you wait maybe a couple of weeks, fully, and then you do it again, and you don't even get sore. You don't get much.
Yeah, that's it. It's a really interesting field, this repeated bout phenomenon. I think it's fair to say that we still don't really understand the mechanisms behind it. There could be a neural aspect. Some of the studies we've done suggest that it's perhaps the connective tissue remodeling or adaptation after the first bout that that might be behind this protection as well. But I think it's an interesting field to pursue, to try to find out what causes the repeated bout effect, because there could be some clues in there as to how we can protect athletes against injury, for example.
That's true. I remember they used to talk about, this is years ago, the susceptible fiber idea that you'd do eccentric. So just so people know at home if they don't know. So when you're contracting and you're shortening the muscle, that's concentric. When the muscle's contracting and it's lengthening, that's eccentric. So we're not talking about just if the muscle lengthens and it's not doing anything. It's contracting and lengthening, and we know that it does more damage. But we also know the muscle can produce the highest forces during eccentric contractions. But so just with this damage, they used to talk about the susceptible fiber business. Yeah. So why don't you just expand on, the weak fibers would drop out, and then therefore you'd be better off next time. I don't know if that's still holding up or what, but yeah.
Yeah, I mean, I suppose a lot of the work that was done on this muscle damage field maybe 20, 30 years ago was looking a lot at the type of damage that I think is repairable without replacement of the muscle fiber.
Okay.
So we had, for example, there was a lot of really nice electron microscopy work done back then, looking at streaming of Z lines, so a loss of the sarcomere structure, particularly at the Z lines, and this idea that some parts of the sarcomere might be more susceptible to damage than the other. I'm not sure that all of those signs of damage, though, would elicit a full repair response. I think maybe they could just be repaired locally without, as I said, the full fiber being replaced. As for the susceptible fiber idea, I'm not sure. I guess with all training, we have to remember the principle of recruitment of motor units. And if you do some exercise for the first time, particularly the eccentric contractions, that's going to be novel for many of those motor units that you recruit. But if you continue to do this exercise, you will recruit additional motor units that are not were not recruited initially. And this is, of course, one of the initial adaptations to resistance training. One of the reasons we get stronger is because we're able to recruit more motor units.
Yes. I think we might get back to that. I'm planning on talking about strength and how much of it is neural and how much is hypertrophy and all that. But before I forget, you were talking about immune cells. So, activation of, pro-inflammatory cytokines and all that's coming in and activating the stem cells or the satellite cells. So it makes me think about, when you do an injury nowadays, it's sort of like quick, rest, ice, compression, elevation, get some anti-inflammatories on board. And, it's like you're trying to stop all this as much as you can. Is there like a danger? Like, should we actually let nature take its course in some ways? Or?
Yeah, it's a good question. And in many cases, I think the answer is that nature does a pretty good job of repairing the tissue itself in the right sequence of events with the right time that it takes. And if you try to interfere with that, then maybe you have some shortcuts that actually might leave the tissue more vulnerable to injury in the future. We've shown just experimentally that if you give healthy young people ibuprofen, one of these non-steroidal anti-inflammatory drugs, and do a bout of eccentric contractions, that the exposure to ibuprofen is going to block the increase in the number of satellite cells in response to the exercise.
Oh.
Okay. So whether it's blocked or delayed, I mean, this is our issue, if you like, when we take biopsies, that we've chosen specific time points, and we have day 2 and day 7. We don't know what happens in between that or after that, but it certainly seems like NSAID seems to block satellite cell activity, and whether that's a direct effect on the satellite cells or an indirect effect, we're not sure. But I think in the clinical situation where you have like a strain injury, for example, there could be an advantage to perhaps dampening the inflammatory response at some point. I mean, you don't want to block it completely. But I think what we know as well is that if you stay in this pro-inflammatory environment, which is the first stage of inflammation, you won't have full tissue repair. So you need to have a shift where the inflammatory cells, particularly macrophages, they change from this pro-inflammatory state to being anti-inflammatory. So they're releasing different cytokines, and this shift is vital for full tissue repair.
Okay, so maybe the way it tends to work out is maybe all right, because you tend to sprain your ankle or something, you race home, you put ice on it and whatever, and then by the time, no, it'd probably be too soon, wouldn't it? By the time people are taking anti-inflammatories, it'd be a few hours later. That would probably be too soon, wouldn't it? You'd want to let it do its thing.
I'm not sure, because I think there's probably also a difference depending on the tissue. So if it's not all soft tissues are the same. So ligaments and tendons, for example, might be different to muscle. And even the definition of what a muscle strain injury is, I think, is not well defined at the moment. I mean, it's based on MRI and ultrasound scans, and there's very little evidence, actually, if any, to show where at the ultrastructural level. So James Tidwell did a study several decades ago now, I think, showing that in the animal, if you pull a muscle to the point of rupture, depending on whether the muscle is contracting or not when you do this pulling, it will separate from the tendon at different places. So in one case, the strain injury is actually a separation of different layers within the tendon. So the muscle is not affected at all.
Okay.
Whereas in the other state, it's actually more towards the muscle. So I think there's still a lot of work to do there to define the actual site of
We should
strain.
let the listeners know that the animal will be anesthetized at the time.
Yeah, sorry, of course, yeah.
You hear about some horror stories with some experiments, but I've done human and animal studies as well, and I think you actually need both. But just thinking about the injuries. So because I was a bit silly, I was talking about spraining your ankle, which is not really, we've been talking about muscle. Spraining your ankle is going to be more ligaments and tendons and things. But if we think about muscle, if that's all right, when you do injure your muscle, obviously it depends on how you've injured it. But if it's, running or something, where does it tend to get injured? Is it the musculus tendinous junction, which I guess we haven't talked about mainly? Or?
Yeah, so the attachment to the tendon again, depends on the anatomy of that muscle and how it's arranged. So whether there's a single point connection, you get to the end of the muscle and the start of the tendon, and you have this junction, what we call the myotendinous junction. Or for most muscles, where it's more like an aponeurosis, where you have this extension of the tendon going up through the muscle. So for these pennate muscles, the fibers are coming in at an angle, and they are attaching to this sheath or layer of tendon, basically. But it's the same connection. So we've started studying this, and you see that it's very interesting, because the ends of the muscle fibers, they express different proteins than the rest of the muscle fiber, both within the cytoplasm, but also the muscle fiber membrane and the basement membrane. One of the best known is specific collagen type, collagen 22, which Manuel Koch from Cologne has shown that this is found at not just this junction, but many tissue junctions throughout the body. So wherever you have two different tissues meeting, it seems they need special proteins to form the connection.
Okay. So just, I guess if we're talking about tissues meeting, we should clarify for people. So a ligament is connecting two bones, and a tendon is connecting like the muscle to a bone. Yeah. So, yeah. And just maybe explain the pennate business as well. Yeah. So, and what kind of muscles are, the more continuous type, so like the Achilles or whatever, compared to one of these pennate type ones. Yeah.
Yeah, so the muscle that we biopsy the most would be the vastus lateralis. So that's one of the 4 quadriceps muscles at the front of the thigh. And even though the thigh muscles themselves are quite long, obviously covering most of the length of your femur, the vastus lateralis, for example, we know that the fibers come in at an angle, like a sort of fishbone type pattern going down through the muscle. So each muscle fiber is only a few centimeters long, compared to, say, the example I used at the hamstrings, where you would have very long muscle fibers.
Okay. So the vastus lateralis, which is the classic one, yeah, your biopsy with cyclists, is more the pennate one, yeah, where they're coming in on an angle. Okay.
Yeah.
All right. And so the injuries then tend to be at that. So I said muscular tendinous. Is it myotendinous? Is that the correct way now?
Yeah, muscular tendinous or myotendinous junction. And you can have injuries at many different locations. So at this aponeurosis, you can have them at that sort of deep inside the muscle. You can also have it more superficial, where the muscle fibers are connecting to the fascia, myofascial injury. You can have them in many different locations. And there's also, I don't have the statistics at my fingertips, but there are also differences as to whether, for example, in the hamstrings, which would be the most often injured in, say, football, there would be differences in whether it's the distal or the proximal part of the muscle that gets injured. In terms of prevalence.
And what is actually causing the injury? Do you know? Is it micro tears, and is it sort of like, is it generally things just rupture and that's it, or is there sort of generally, I don't know if you know this, I'm just, is it like there's already something going on, and then it pops, sort of thing? You know what I mean?
Yeah, it's interesting. So whether it's I think if we knew for sure, we would be better at preventing them. I think the fact is that I think for football hamstring injuries, for example, I think you're looking at about 1 in 10 players per season getting a hamstring injury. And for some of those, that will be the end of the career, because we know that even though the tissue will heal, we know that the biggest risk factor for getting a muscle injury is having had a muscle injury previously. And it's a bit problematic, because it's also been estimated that about 80% of all of these re-injuries occur at the same site as the initial injury.
Oh, okay.
So I think that, at least to me, that says that whatever we're doing in terms of rehabilitation and most of these people are athletes, and they're getting the best rehabilitation, and it's very structured and targeted rehabilitation exercises under supervision, and there's very good compliance because these individuals are highly motivated, and even in that situation there's still a very high rate of re-injury. And to me, from going back to the cell types involved in the muscle adaptation, I'm quite keen to find out what's going on there. Why is the tissue not healing completely? Because that's what it seems like, right? That if you get a re-injury in the same site as the initial one, there must be a problem with the initial healing process. And whether there must be something we can do differently to improve.
I guess everyone's going to be different, but sometimes people tend to go back too soon, right? There's a bit of pressure to come back. So I guess it might be different if they get that same injury straight away versus, months down the road or whatever.
Yeah, absolutely. And I think most of these re-injuries occur within, I can't remember, but it's definitely earlier than later. Okay. So the ones who wait longer, perhaps. And I don't know if anyone has looked at that. For example, the pressure that some of these players are under to get back in the game, depending on when they get injured in the season, for example.
So what about, again, this is quite a lot of practical stuff, so we haven't got that much in the nitty-gritty, but just do you know, like, the best way, because it seems like things get more and more aggressive. It used to be just rest when you get injured, and then it was like do some strengthening and some stretching, and now it's sort of like get as soon as you can start mobilizing the area or whatever. Do you have a feel for, and I know we were laughing a bit earlier before we came on air about nowadays with, chronic tendon injuries, you can actually, like, I was saying I felt like sticking a fork in my Achilles at one stage, and you said that might have actually worked. So do you want to just explain a bit there?
I think in all of these sort of areas of research where, sometimes some of the treatments develop where the money is, right? And often ahead of the science. So, for example, one example would be injection of this PRP, platelet-rich plasma. So you take a blood sample and you concentrate it with all the good stuff and inject it into the site of injury. And a lot of clubs were doing this. And there were even a couple of publications on it. And then people started doing, randomized controlled placebo trials where actually there was a group that had an injection of just saline, and there's no effect of PRP. So it's, I think if some of the better clubs start doing it, then everyone wants to start doing it. And I think the talking about sticking a fork in your Achilles, painful tendon, probably make it more painful. But I think some of that, there was this dry needling idea as well. And there was a study done here at Institute of Sports Medicine where they just injected a needle without injecting anything, just inserted the needle into the tendon, and that in itself will stimulate a lot of activity of these cells. So there could be something in it. It wasn't a study that showed that you had improved repair, but I think part of the problem that tendon has that muscle doesn't have is that stimulating repair of tendon seems to be difficult. It's difficult to activate the cells that are in there.
Yeah. It seems almost like when you've got this chronic, like Achilles injury or something, that it just stays the same. It doesn't get worse, doesn't get better, and you feel like you've got to change something. So that might be the idea, that when you start dry needling or sticking a fork in there or something.
Or do something different. Yeah, there was a study done here. I came to Copenhagen in 2004, and I think shortly after that, there was a PhD student here, Mads Kongsgaard, and together with Peter Magnussen, they did a study where they thought they would try something a little bit perhaps counterintuitive for tendinopathy. So they got these people to do eccentric loading of the tendons, and it was slow and progressive, and they allowed some pain, which I think was different to earlier studies, And the interesting thing was that this seemed to improve not just in the short term, but also in the long run, the pathology of these individuals. So I think, I mean, I don't know if sticking a fork in your tendon is the right thing to do, but I think sometimes you need to do something that's maybe a bit counterintuitive.
Forcing it.
Kickstart some of these processes.
Well, because it almost like maybe it becomes a new baseline or the new sort of set point or something, that this chronically inflamed or something. Because I've got a mate as well who told me that he had Achilles tendon problems, and he had heard about someone who said, just operate on me, doctor, and they said, no, it's not bad enough yet. So he said, I'm just going to work on this. I'm just going to work it out like crazy, do all these crazy squats and whatever. So it ruptures, and then the guy will stitch it back together and whatever. And it ended up just getting better. And then my mate did the same thing. He just went all these crazy squats. And I'm not suggesting people necessarily do it at home. And I know you said progressive or whatever, but he was literally just trying to like bust it, and it actually got better. So it's a little bit counterintuitive, but
I think there's a lot we still don't understand. And certainly for tendinopathies, I mean, this is one of the main reasons why people stop at high level sport. It's because of failure to get over their tendinopathy.
All right. Now, I know you've done aging research, and I couldn't help thinking when you said the most likelihood of having an injury is having a previous injury. So naturally, the older you get, you've got more previous injuries, shouldn't you keep exercising?
Yeah, but aging in itself actually would be one of the next risk factors. I don't remember the order of the importance of these risk factors, but aging is certainly up there, that even if you haven't had an injury before, the older you are, the greater your risk of a muscle injury.
Yeah. So what's actually happening there then? So one of the things I was also noting that you've done stuff to do with innervation of muscle. And so one of the things you tend to think about is, as you get older, is it correct you tend to lose your fast fibers more than your slow? Is that correct? And so you have less motor units? And
I think there might be a difference here between animal and human. And it might look like in humans that you lose fast motor units at one point in life, and that might happen a little bit earlier than the type 1s, the slow motor units. Certainly you see in older people what we call fiber type grouping. So if you take a biopsy and look at a cross section with several hundred fibers, in younger people, the type 1, type 2 distribution would look a bit random. Whereas in older people, you tend to have clusters of, maybe 20 of one fiber type. And there are several potential explanations for that, but one of them is certainly that you have lost the other fiber type. And as to how that happens, I think, to be fair, it's still quite an open question. So there was one study in 1977, I think, on human cadavers, Tomlinson, showing that, they actually counted the number of motor neurons in the lumbar region of human spines, and they could show that there was a decrease in the number of motor neurons throughout life, or from middle age, sort of age 40, that this number would start to decrease. And that would fit quite well with some of the work that Jan Lexell did in the 80s, I think, where again with human cadavers, he took the whole vastus lateralis and cut sections of these muscles of giant microscope slide, and basically counted all the muscle fibers. And he had samples from people of all ages across the lifespan. And similar to the number of motor neurons declining with age, he also saw a decrease in the number of muscle fibers.
Because I thought maybe the way they used to sort of explain it, I thought was that, you're losing motor units. Yes. So just to clarify for people, you talked about motor neurons. So coming from the spine, and then they innervate the muscle and they kind of spread out. So you get, you know, like, it's not like one nerve is getting one fiber, they spread out. And as you said, you tend to get, it's almost like random in young people. And then as you get older, if you've got less motor units, you tend to have more bunching of different types. My feeling though was you weren't actually losing the muscle fiber number, you were just having, you know, the same number of fibers but less motor units. So, you know, it's almost like more crude control. Is that not right? You're actually losing muscle fibers.
Well, I think both are correct actually, because let's imagine that one motor neuron dies.
Mm-hmm.
And let's go back to the quadriceps muscle we were talking about earlier. So some of those larger muscles would have maybe, I mean, they would have thousands of muscle fibers in one motor unit.
Right.
So one motor neuron is going to connect to thousands of muscle fibers within the same muscle. So if one motor neuron dies, then let's say there are 7,000 muscle fibers in that motor unit. Mm-hmm. They are left without the ability to contract. They're not getting any signal to contract. And potentially they will eventually atrophy, they will get smaller, and they will die. But we know that neighboring, and this is maybe what you're referring to, that neighboring motor neurons then will pick up the signals coming from these muscle fibers. It's like they send out these, they start to express proteins. This is one of the things we've been looking at in the denervation studies, that these muscle fibers, once they lose connection with a motor neuron, they start to express proteins that attract neurons.
Nice.
So some of the neighboring motor neurons then would, their axons would branch and sprout, and you have what's called reinnervation. So those muscle fibers then would join another motor unit, and that motor unit, as you say, then would become larger. But I guess at some point there's a limit to how many muscle fibers a motor unit can adopt, if you like. And I suppose that the rescue is never 100%. So out of these 7,000 muscle fibers, I don't think 100% of them would be rescued.
Okay, that makes sense. Yeah, I was thinking they were all getting rescued, but it makes sense. So as you're getting older, you get more injuries and you recover slower and more likely to get re-injured, do you know? And do we know why that is? Is it because they got less fibers, less motor units? I guess that means you're not as, I don't know, maybe your contractions aren't as well controlled, or.
Yeah, I suppose it depends a lot on perhaps the exercise history of these individuals. So I think I read recently that the unconditioned male in midlife is at greatest risk of muscle strain injury. Okay. I don't know if that reflects a midlife crisis and trying to keep up with life and fitting in short, intense exercise periods, trying to compensate for regular training. I don't think there are any shortcuts when it comes to exercise training, unfortunately. But we know that even the people who don't train and then take up some exercise and get injured, that people who try to maintain their training throughout life, I mean, you see this in for example, the Masters athletics championships, where you have age group 90 to 94 and new world records being set for the shot put or the 100 meters or whatever within each of these age categories. It's fascinating. But these individuals have, most of them have been training all of their life. They've been physically active. And they try to, and they keep up their training volume, many of them, and still they report that they have, they seem to get injured more frequently. And why that is not investigated at all, really. Whether it's a consequence of fewer motor units, as you say. And I mean, what we do know is that these muscle strains, they tend to occur during very high force, explosive movements.
Okay. Yep.
Like an all-out sprint, kicking a ball. And often it's in a state where like in the competition setting, actually, which can be difficult to replicate in a training situation where people are really pushing themselves
Right. Yep.
above maximum capacity almost. So whether something happens with aging to the composition of the connective tissue, that's entirely possible, that you have changes in the different types of collagen or the proportions, either at the myotendinous junction or throughout the muscle. We really don't know.
That's true. I mean, I guess with aging, you lose elasticity of everything, right? The skin, the lung parenchyma, and all sorts of things. So I guess that's what's happening. Now, I saw you had a paper. Where is it now? I've got it written here. Impact of high resistance training in older individuals on myofiber innervation. Now, I don't know what you got there, but it got me thinking, you know how you lose these these motor units as you age. Can you actually slow that? Can you change that, or is that fixed? And you know, if you're doing this resistance training, what does that affect on innervation?
Yeah, I don't know. I have had a PhD student who just got his PhD degree a couple of months ago, Casper Sundbro. He's the first author on those papers. And it's funny how you get into these fields. I hadn't really considered it much before. It was because we'd been looking at, we had muscle biopsies from masters athletes, so endurance runners who'd been running marathons. Many of them had run more than 100 marathons in their lifetime.
Wow.
And I looked at satellite cells, muscle fiber size, a couple of other things. It was a big study. There were several people involved here from the Institute. And one of the things one of the reviewers asked for when we were getting this paper published was whether there was any sign of regeneration of the muscle fibers in the athletes, if that could explain satellite cell number or anything. So I did a staining for a classic regeneration marker, neonatal myosin, which you don't see in adults. It's only expressed during development of the first muscles before birth. And there were quite a lot of these fibers in the athlete biopsies. But the interesting thing was we also had a group of older age-matched controls who were not athletes, and they had just as many.
Oh, okay. Well, that's why you need a control, yeah? Okay.
Yeah, exactly. And it turns out that actually it's been shown, Stefano Schiaffino showed quite a long time ago now, that if you denervate an animal muscle, like cut the nerve, so the whole muscle has no neural input, then all the fibers will express neonatal myosin. Yeah. So it is a regenerating marker, but it is also definitely a marker of denervated muscle fibers. So that one observation really got me thinking. And at the time, Casper had come and asked if he could do a master's with us. And I sort of said, well, you know, yeah, we have this thing that we think might be worth pursuing. And I suppose that's one of the advantages of a master's thesis, that you can play around a little bit more, right? You don't have to have a full project carved in stone. So, and he was the type who was willing to go into something open like that, and started looking at some of the other samples we had. And we could see there were quite a lot of these fibers in some of the older populations that we've studied. So we started to look at some of the other markers of denervated muscle fibers using sectioning at the microscope, and a lot of fiber counting and positive and negative fibers. And this was quite a consistent finding. So then Casper went on to do a PhD, and a whole PhD came out of this observation, actually. And his main study was then to see exactly what you were asking. Is there something we can do to slow down or even prevent the loss of muscle fibers? So we say it's never too late to take up exercise, and that's true. We should, I mean, anyone at any age will benefit in so many ways from taking up exercise. There's no question about that. And even if they're very old and start, for example, resistance training, their muscle fibers will grow in size, they will increase in strength, and there'll be a lot of other benefits too. But I think that one of the key points here is that those muscle fibers that are lost, we can never get them back.
Yeah.
So, and we start losing muscle fibers probably from the age of, I'm nearly 50, somewhere in 40s, 50s. And the potential to prevent that, I think exercise is maybe the only thing actually that is, I mean, the most obvious thing now to consider would be that exercise could do this. So Casper, for his PhD, he recruited lifelong exercisers. So we tried to get not the long distance runners we'd studied before, because they're kind of strange in a lot of ways. They have a lot of type 1 muscle fibers. So everything we study at the biopsy level tends to be different anyway because of this larger.
So they're hard to match.
Fibers. It's hard to match, right? And we wanted to go for thinking along the lines of where we started, that perhaps the type 2 fibers, the type 2 motor units, are more susceptible to loss during aging.
Right.
So we wanted to find people who would be recruiting their type 2 fibers continuously throughout life. And it wasn't that easy, actually. We got in touch with a few people who've been doing resistance training, doing weights basically, but a lot of them were not continuing when at the age of 60 when we wanted them. So we ended up with this mixed bag of people doing a little bit of everything, going to the gym, maybe a bit of running, a bit of rowing, badminton.
Which is probably good, right? I mean, it's a bit harder to control, but it's good for them to be doing a mixture of things.
Oh, absolutely. And in the end, I think I call them the mere mortals, because, you know, this is not, and that's the other thing about studying masters athletes. It's all well and good, but it's a very small proportion of the population that it actually represents. And anyway, what we found was that it's not clear. I mean, we study something and we see something at the microscope or gene expression level, and we interpret it, right? But our interpretation of this, of these findings is that this type of exercise preserves, first of all, muscle stem cell number. So they had not lost as many satellite cells as the older inactive people. And there were signs in their muscle biopsies that that perhaps this lifelong approach to exercise was leaving them with fewer denervated muscle fibers.
Wow, so they should end up with more fibers in the end. Total.
Yeah, or at least they might slow down the loss of muscle fibers. So we tried with a group of older men then to do 3 months of heavy resistance exercise to see if that will be enough to perhaps reverse some of the, and you can't get back fibers that have died, but if you have fibers that have become denervated, then maybe the whole exercise environment, so not just the muscle fibers, but also satellite cells, you change the number of immune cells in there. So the whole environment in there will be different in someone who's exercising compared to someone who's not. And perhaps that environment is more conducive to re-innervation, rescuing some of these lost fibers. And we could see after 8 weeks of exercise, there were some signs that this might be the case, actually. So again, I think while we say that it's never too late to take up exercise, I think that's probably not true if you're talking about preserving muscle fiber number.
Yeah, so to make sure people are clear again, so what we're talking about, remember we talked about you lose a motor unit and then you might have 7,000 fibers. and some of them are going to die, and some of them are going to be taken up by adjacent motor units, and you know, interwoven. And you're talking about that maybe when you lose a motor unit, if you're exercising, then maybe more of those won't die and will be taken up by other motor units. Yeah.
Yeah, and the other possibility is that some of these factors produced by the exercising muscle will be fed back up through the motor neuron to the level of the spinal cord to actually prevent loss of motor unit
loss. Wow. Okay.
I mean, that would be fantastic. But again, to be fair, I think the evidence for loss of motor neurons with age it's not that strong.
Okay, so what you need to do is go back with, you know, these said ages ago that guy went and got cadavers of dead people and looked at, so, you know, you should try and get a hold of, well, it's easy to get a hold of them when they're dead, but if you can actually get access to them, you know, people that have never exercised and count how many muscle cells they have, people that have exercised their whole lives, strength trained, endurance trained. I mean, it's a big study, and I'm sure there's all sorts of hoops to jump through, but that would be pretty interesting.
Yeah, and one of the other things, going back to that master's athlete study that we did, where we spotted these denervated muscle fibers for the first time, one of the interesting things from that was that we measure, you know, there are different ways of measuring how much muscle you have. So we had MRI, where you basically get a scan cross-sectionally through the whole thigh, and you get to see how big the whole muscles are. And we'd also measured the size of individual muscle fibers from the biopsies. And we could see that the athletes had larger total muscle cross-sectional area of their thighs, but their muscle fibers were not any bigger than those of the controls. So, and there can be many factors. There's pennation angle in pennate muscle and other things that can contribute to the total muscle size of an MRI image. But it's tempting to suggest that, you know, it's their exercise habits that have maintained muscle fiber number, so that they actually have more muscle fibers than the untrained controls. But again, it's a cross-sectional study, and this is one of the issues we have with humans and studying aging. We also can't rule out that those people were born with more muscle fibers to start with.
Right. It is surprising though that you don't have you know, normally we talk about hypertrophy with strength training, hypertrophy of fibers rather than hyperplasia, which is more muscle fibers. But I know you're not saying that, because what you're saying is rather than, you know, strength training causing hypertrophy, which is increase in each cell plus more cell numbers, which I think we know doesn't happen. You're talking about you're just not losing the cells. Yeah. I
mean, I think it's obviously hard to prove, right, whether you can get more muscle fibers in human. And maybe just to go back to clarify what I was saying at the beginning about satellite cells forming new muscle fibers, they replace damaged fibers. I don't think we get new muscle fibers. And you know, there are some reports which again are fascinating for people like me who see a lot of weird things at the microscope, but where you see fiber splitting or branching, or these reports from older bodybuilders. And I think this has been some of the fuel for the fire of people thinking that you can get more muscle fibers in human muscle. I don't think that happens. I think these are simply reflecting the process of muscle repair, actually.
All right, well, that's great. So I wanted to ask you a different sort of question I've been asking people lately, which is, you know, it's easy for people to listen and think, oh, okay, every study you've ever done and every study I've ever done, it works. You know, you get what you want, you get what you planned on, or whatever. I've just been asking people like to flesh that out a bit. I think it might be good for up and coming students to know that you don't always get what you think you're going to get. And I said in the last podcast, I think that probably 70% of the time I get different to what I actually thought I was going to get. It doesn't mean it's bad. It's just different. And sometimes it's interesting and you go with that. Do you have anything pop? I'm hitting you cold here, but any thoughts on challenges
no,
you've
I
had?
am more than 70%, I think, of the studies that I have designed have not turned out, I have not been able to confirm the hypothesis I had.
Yeah.
And I think I've had a lot of negative, what we call negative findings, and this is something, it's a bit difficult to, I mean, some people think that a negative finding is like a failed experiment.
No.
It's not, and
No.
it should still be published. And I think that's been one of the frustrating things for me, that it definitely seems that these negative findings, journals are not as interested in them. And it's, personally for me, I find that very disappointing, and I find it very frustrating. And I think looking back, I've probably been a bit too naive in terms of thinking that some of these exciting animal findings would transfer to humans. So I've set up a couple of human trials based on, we had one on blood pressure lowering medication, at the angiotensin 2 type 1 receptor blocker. Some of them go by the name losartan. There was a bit of hype about it and some muscle animal literature a few years back, suggesting that it could slow down muscle atrophy, increase IGF-1 signaling in the muscle, reduce TGF-beta signaling. Like, all of this sounds fantastic, and imagine
all the stuff that should have a positive effect on muscle mass. Yeah.
So we set up a study in humans to do resistance training in old people, and we gave them this. It was a randomized controlled trial. We had 80 people involved. We found nothing. The control group and the drug group had exactly the same outcome, apart from blood pressure, although we did see a small decrease even.
Well, there you go. So you're able to get reproducible findings.
Oh, but you feel a little bit, oh, why did we do this? You know, I mean, it's a huge effort. And I don't know if it's, and I've since learned that the renin-angiotensin system is more complex in humans.
But I totally agree with you. I've talked about this before on the podcast, but this so-called negative findings or whatever, I think it's just, you know, if you hadn't done the study, then you wouldn't know. And often it's really important. It's just as important to not continue down that line or whatever.
And to let other people know that don't waste your time doing this.
Exactly.
I mean, it's,
And it means something, right? The fact that this didn't affect this or whatever means something. And often you end up going, oh, well, that's interesting. And you'll go off. You know, the main thing is you keep an open mind, because you go, oh, hang on, that's actually really interesting. Let's follow that further.
Yeah, and I think I've probably done that on 2 occasions. One was the example I gave earlier, where we found these neonatal myosin positive fibers in the older muscle, which turned out to be denervated, and that is turned out to be a huge focus for me currently and in the future. And the other thing was even looking for a model of muscle damage. That's also been quite a bizarre story for me. So in my PhD, we did the classic 200 voluntary eccentric contractions, and I thought I'd be studying muscle damage, and I was really looking for, you know, we had high creatine kinase, 20, 30, 40,000 units per liter, which is really high. And they were very sore, these people. They couldn't walk down the stairs and get their trousers on and all this. I mean, this was the response. So and that was my first experience of working with human biopsies. And I remember being so disappointed looking in the microscope, not a single damaged fiber in any of them. Wow. And we had taken several time points, day 4 and day 8 or something, nothing. And then when I moved to Copenhagen, there was an Australian postdoc, Regina Kramery, who had done a study published in Journal of Physiology, where she took these young individuals and she had them do the classic voluntary eccentric contractions with one leg, and with the other leg she used electrical stimulation. And when she left, there were a lot of her biopsies in the freezer, and it was amazing to look at. I mean, she showed a couple of illustrations in the paper, but it was clear to me, if I wanted to study muscle repair I had to have damage in the first place. So that was the reason I went down the electrical stimulation.
But that's pretty weird though, right? Because that's actually not as physiological. So the way you were doing it originally is more physiological, but you're not actually seeing the damage.
And that's also one of the things. It's kind of come full circle now, because I was also really tired of, and I think if you study satellite cells in muscle, of course it's muscle, most of the reviewers we would get would be people working with mouse. There were not that many people working with human muscle at that time in the context of satellite cells. And I was so tired of getting comments like, oh, but there's no damage, and you need to have real damage if you want to study regeneration. But you're right, the electrical stimulation model is not physiological. The only physiological situation it would be representative for is something like CrossFit. I don't know, do you have that in Australia?
Yeah.
Yeah. So, I mean, where you have people doing these assisted lifts during the concentric phase, and then they're doing the eccentric phase, and you have people admitted to hospital because they've gone overboard. And there they have what we see with electrical stimulation. Wow. And it's quite interesting now that just to finish that's another sort of personal disappointment of mine. When I finally had this model of real regeneration, and I submitted it to a big journal, and it was rejected. The comment, one of the reviewers' comments was, "This is known for the mouse."
Oh my gosh. Yeah, well, what I was thinking earlier was the models they use, right? So the people reading about mice, they'd be used to these cytotoxins where you inject something that destroys like the whole muscle, or as you said earlier, they cut the tendon or something. How physiological is that?
Yeah, no, it's not either. And to be honest, I think what we have with the electrical stimulation is a model that doesn't represent real life injuries, but it is a model where we can study activation of satellite cells and the interaction with fibroblasts and immune cells in a sterile environment that is definitely not cell culture, right? We're studying it in vivo. And if you can administer drugs, for example, NSAIDs, some other drugs, either locally directly into the muscle or systemically, it's a very good model for studying some of these cell interactions. So, but it is a model, and it's not a strain injury model.
It's really weird how you said that when you do the voluntary eccentric contractions, you get these really high CKs. So for people, that creatine kinase is meant to be in the muscle, and if you've damaged the muscle membrane, it leaks into the blood, and you can measure it. And that's how people, But you're saying you couldn't pick up the CK. The creatine kinase is really elevated, but you couldn't pick up any muscle damage. So how's it getting there?
Yeah, I don't know. There could be some small
permeability.
There could be some and again, it's a level of detection. I mean, I didn't do electron microscopy in that study, and there could have been a lot of streaming of Z lines, sort of ultrastructural damage, but we didn't see infiltration of immune cells, for example, into muscle fibers. So yeah, I don't know how the creatine kinase gets out.
All right. Well, this has been really interesting. I think we've covered a fair bit. Is there anything that we haven't sort of covered that you wanted to bring up, or some sort of hot finding you want to mention, or anything like that? Or what do you reckon?
No, I don't think so. I think we've covered most things.
We've covered a fair bit of ground, actually.
Yeah.
Yeah. So it's been great. So thank you very much for coming on.
Yeah, you're welcome. Thanks for the invitation.
Well, great. Okay. See you around. Bye-bye.
Yeah. Bye.
Bye.